Hey guys, so in this screencast today, what I'm going to show you how to use is a really powerful tool from a biological supplier called New England Biolabs. It's going to allow us to predict what's going to happen with any sort of restriction digest. This is a tool I expect you to be able to use. So get familiar with the website. Also, what I'd like you to do is, as you're watching the video, you should have your lab notebook with you. Be sure to write down a detailed procedure on how to use this online tool in your lab notebook. This is something that you're going to want to have for reference in some of the projects that you're doing, but also in the assessment that we have. This is something that you're going to want to use in order to make predictions. So the website here is really simple. It's just neb.com. That stands for New England Biolabs. So if you search it on Google, you'll get the same thing. And here's their website where you order all sorts of stuff. Anybody who's doing any sort of research in molecular biology or biochemistry, this is a iGEM. This is a really popular website and a very popular company to order restriction enzymes from. They do a really good job with their restriction enzymes and all sorts of molecular biology tools. If you go to tools and resources right under here, you'll see that they have this online tool called NEB Cutter, and that's what we're going to be using today. This is the tool that I want you to be able to use, is NEB Cutter. So if you click on that, it's going to open a brand new page. That looks like this, not a super fancy page, sort of bare bones. It doesn't need to be fancy because it does its job so well. So if you look over here in this part that says standard sequences, I want to show you two regions here, okay? Because one might be useful for you later. The first one is called plasmid vectors. These plasmids are what you can use to insert into bacteria in order to have them express some sort of recombinant DNA. So maybe something like GFP. In which you can look right over here and you have a ton of different plasmids that you could click on. Those are circular. So we'd have to do something a little bit different here, which I'll show you in a second. What we're actually focusing on is viral DNA. It's a phage DNA. You're going to cut something called lambda DNA. It's a bacteriophage. So I'm going to click on lambda right there, and that's my standard sequence. So that already knows the DNA sequence. I think it's 48,502 base pairs of lambda DNA. It already knows it, so we've got it loaded in there. Okay, the sequence is going to be linear, so you don't have to worry about that. So my next step is just to hit submit, and what's going to pop up here is lambda DNA. All the way from number 1 to 48,502, it's got a little line there representing where all these cuts may happen because of different restriction enzymes. What I'd like you to go through and do next then is go right over to this, which is a custom digest, which is going to allow me to pick what restriction enzyme I'd actually like to use, and up pops all of New England Biolabs restriction enzymes. Notice how many different flavors of restriction enzyme there are. Let's just go ahead and pick one. I'm going to go scroll down. They're all in alphabetical order to one of the enzymes you're actually going to use in lab, which is called PST1, which is right here. Here's the recognition site, so it's going to cut in between. The sequence is going to be, the recognition sequence is CTG, CAG, and it's going to cut in between C and T and the A and the G there, and it should do that 28 times when mixed in with some lambda DNA. So I'm just going to click on this little box, and then I'm going to hit right down here, I'm going to hit digest, and up pops all the spots that PST1 will actually digest the lambda DNA. Okay? Let's go right down over to this part that says list. Now what this is good for is to show me what are the expected lengths of the sequences, and that's something I'm going to ask you to be able to predict. So this is a useful tool for you. Notice you got a couple of tables. One table over here goes from the first cut, which is going to give you 11,497 base pair length fragment. This one goes kind of in the opposite direction. You can look at either one. But here's your 29 total fragments that you should get from the cut, and then here all in a row is where you have the length in base pairs. So the first cut's going to be the longest, and then this is what we'd actually expect to see in terms of base pair length on a gel, if we were going to run this on a gel, which you will. But here's what's really cool about NEB Cutter, is you can actually go view the gel. Like, what would that look like? So I can hit right here, view gel. And what I want to select is the correct percentage of agarose, which I can do right up here. I can select the marker. We want unmethylated DNA. Remember, like we talked about in class, if something's methylated, the restriction enzymes can't really get to the recognition site, so they're not going to cut. So this DNA is unmethylated. You guys are going to run this on a 1% agarose gel, which you've already made. You're going to click on that right there. And notice what happens is these bands don't travel as far because the 1% is thicker than the 0.7% agarose. And then what we also need is a marker, some sort of standard of comparison. So your marker for this lab is actually going to be lambda that's had a HIND3 digest added to it. So HIND3 is another restriction enzyme that's cut lambda. And we know now all the lengths that we should be able to see, be able to visualize with that specific cut. So if you hover over this, you can see that first cut should be 23,130 base pairs long. Now what I could figure out based on that marker and the second marker, which is 9,416 base pairs, I can figure out how long that base pair should actually be. This is also going to give me the answer right there. It's 11,497. That's the first one that we should see, the first band. And notice that matches up with the first cut right here. So our longest band, the biggest band in terms of base pair length, is up towards the top because the big ones don't travel quite as far as the little ones do. If I scroll down, you see this one is really, really small. It's only 15 base pairs. There's no way that we're probably going to be able to see that actually on a gel. There it is, just 15 base pairs. It's the smallest one. It travels the furthest. Okay, so what you can do is actually print this off. You can go right up here and hit print or do a screenshot of it. And what that's going to allow you to do is actually make a prediction of what that digest should look like with any sort of specific restriction enzyme. Okay, so this is a great tool for being able to make those predictions and then actually run the lab with your enzymes and then see, do you get the expected results? Okay, if you guys have any questions, please let me know. But that's basically it for the NAB cutter. Good luck.